Hi friends, in this video we are going to learn about the internal structure of kidney. To understand the internal structure of kidney, we are supposed to take its longitudinal section. It means we have to cut the kidney longitudinally into two halves so that we can observe its internal structure. Okay. Then the outer surface of the kidney is convex in shape and the inner side of the kidney is concave in shape. Okay, and the peripheral region of the kidney is this region. This is known as peripheral region. It is dark reddish brown in color, and the innermost area of the kidney is light brown in color, or light reddish in color or pinkish in color. This is known as a medulla. Okay, so the outer surface is dark brownish in color. This is known as cortex, and this region is inner region is known as a, here in this on the concave side of the kidney you can find a, so this is known as fissure region. So what is the specialty of this fissure region? This is also known as hilus or hilum. Okay, then here you can see that renal artery enters into the kidney, renal vein comes out of the kidney, and ureter comes out of the kidney. Okay, so this is the ureter. Let me show you in the excretory system. You can see here, see, this is known as renal artery, means this is the dorsal aorta. So this, from this dorsal aorta, one branch of blood vessel is divided and it is entered into kidney. This is only known as a renal artery, because renal is a word related to kidneys. Okay, so renal artery enters into the kidney, renal vein comes out of the kidney, and ureter comes out of the kidney and runs downwards and opens into a bladder. Okay, so in this excretory system you can see here. Once again, coming to the internal structure of kidney, you can see here this peripheral region of the kidney, means this outer peripheral region. This is known as cortex. You can see here cortex, and the inner region is known as medulla. So what is the speciality of this outer region and inner region? Here in this cortex region, cortex is dark reddish brown in color, and the inner medulla is light or pale brown in color. Okay, so here in this medulla you can see some parts known as calyces. These are like flower calyx like structures. That's why these are known as calyces. What is the speciality of calyces? Calyces represents the location of the functional units of the kidney known as nephrons. Okay, so nephrons are located in these calyces regions. So how many nephrons are there in each kidney? In each kidney there are 1.3 to 1.8 million nephrons are there in each kidney. Okay, so you can see here this region is known as hilum. I told you, in this hilum you can find that renal artery enters the kidney, renal vein comes out of the kidney, and ureter comes out of the kidney. So when renal artery enters the kidney, it will be divided into for example, in this kidney, if there are 1.3 million nephrons are there, I mean the lowest number, so 1.3 million nephrons are there in this kidney. This renal artery, after entering into the kidney, will be divided into 1.3 million branches. So after that, each branch enters into one nephron. Okay, so as I told you, nephrons are the structural and functional units of the kidneys. Okay, so there are many nephrons in the kidney. As I told you, 1.3 to 1.8 million nephrons are there in each kidney, and the kidney function depends upon the nephrons function. So if all those many nephrons are functioning well, that indicates the proper functioning of the kidney. If any of the nephrons fails to function, then that is a diseased kidney, we can say. Okay, so because of any reason, it might be because of some bacterial infections, or it may be because of chemicals, the higher percentage of chemicals in the body. So like that, they may damage the kidneys, so here you can see this is the location. Medulla is the location of nephrons. Okay, so now after entering into the kidney, this renal artery, it divides into so many branches known as renal arterioles. Okay, so renal arterioles also, here you can see how they enter into the each nephron. I'll tell you. To know about the functioning of the kidney, we need to learn about the structure of nephron, because this type of nephrons, how many nephrons are there in each kidney? 1.3 to 1.8 million nephrons are there in each kidney. Okay, so before learning the functioning of the nephron, to learn the functioning of the nephron, we have to learn about the structure of the nephron. Then only we can easily understand how it functions and how it plays its role in the functioning of the kidney. Okay, here you can see this is the structure of nephron. So I told you this renal artery, when enters into the kidney, this is divided into 1.3 million nephrons. If there are 1.8 million nephrons are there, it will be divided into, this renal artery will be divided into 1.8 million nephrons, and each branch enters into every nephron. Okay, each nephron. Okay, so like this you can see that renal artery is divided into small branch, and this is known as efferent arteriole. See, this branch, larger branch, is divided again into smaller branch. So this is known as efferent arteriole. Why it is called as arteriole but not artery? Okay, arteriole means when we call arteries when as arterioles, when those are in microscopic in size. Okay, so this artery which enters into nephron is known as efferent arteriole. This small one is known as efferent arteriole. Okay, so you can see here one more one is coming out, that is known as efferent arteriole. Okay, so before into its function, so let me teach you about the structure of the nephron. You can see here this cup-like structure, okay, known as Bowman's capsule. This Bowman's capsule along with this glomerulus is known as Malpighian body. You can see here Malpighian body. So this total region, Bowman's capsule as well as glomerulus, this region is known as Malpighian body. Okay, so how this glomerulus is formed, when this afferent arteriole enters into Bowman's capsule, it will be still divided into so many branches and forms into a group of blood vessels known as a glomerulus. Okay, it means through these blood vessels, this blood used to flow. Okay, so for the proper filtration. Okay, you can see here now, this is the whole nephron is divided into two major parts. One is Malpighian body. This is what Malpighian body, and the remaining part is tubular body. See, this is all tubular body. This is known as renal tubule. Okay, renal tubule, Malpighian body and renal tubule. Okay, so in this Malpighian body, what all process takes place? Let us see. Okay, see here, this afferent arteriole is bringing the blood into the glomerulus. Glomerulus is nothing but, I told you, it's a network of capillaries which are very, very minute. Okay, which are these capillaries, the walls of the capillaries are single celled in thickness. Okay, so that's why through this filtration can be very perfectly done. Then here, so what all can be filtrated here? Let us see. So when this afferent arteriole is bringing the blood into the glomerulus, this blood is loaded with waste materials, I mean unwanted substances, which are nitrogenous substances, which is having oxygenated blood. The blood which is coming into the coming through the efferent arteriole is oxygenated blood but loaded with impurities which has to be filtrated. Okay, so now it passes through this glomerulus, I mean network of glomerulus. Those are nothing but very minute capillaries, as I said, and blood when passing through these, it will be filtered, and nitrogenous substances and some other unwanted substances will be filtered here, and this all filtrate will be gathered in this cup-like structure known as Bowman's capsule. Okay, so after completion of filtration, the blood comes out again into the another blood vessel. Again, these all glomerulus can be again formed into, I mean reformed into blood vessel. This is known blood arteriole. This is known as efferent arteriole. Okay, so you can see here efferent arteriole is coming out. Ring arteriole is known as efferent arteriole. The coming out arteriole is known as efferent arteriole. Okay, so now the filtrate is now entered into this Bowman's capsule. So now from this Bowman's capsule, filtrate enters into, I mean urinary filtrate enters into the tubular part. Next part is known as the tubular part. Okay, so here the base of the Bowman's capsule is like it's like it is having podocyte cells. Special cells are present here. Podocyte cells. It means in between the cells there is a gap. I mean, I'll show you. For example, if these are the cells, okay, you can see here in between the cells there are spaces. Actually, in tissues, these spaces are filled with intercellular space is filled with fluid. It may be lymph or else intracellular fluid. Okay, but in this Bowman's capsule cells arrangement is like this only, but the gaps are still maintained. Okay, so through these gaps filtrate can be entered, okay, into the tubular part. I hope you understood. It's like this type of cells which are there in Bowman's capsule. At the base of the Bowman's capsule are known as podocyte cells. Okay, podocyte cells, those are somewhat special cells here. So next, now through these podocyte cells, the gaps in between the podocyte cells, the filtrate now enters into this tubular part. Okay, this tubular part is known as renal tubule. This renal tubule is divided into three parts. Okay, so first part is known as proximal convoluted tubule. The meaning of proximal is, proximal meaning is beginning. So in this tubular part, in this renal tubule, the beginning part is known as proximal part. And why it is called as convoluted, so you can see here it is able to bend, right? So convolutions you can see here. That's why it is known as proximal convoluted tubule. Why it is called as tubule? Because it is in the microscopic level. So it is not called as tube. It is called as tubule because it is in the microscopic level. Okay. This is known as PCT. Shortly it can be called as PCT. It means proximal convoluted tubule. So now filtrate entered into proximal convoluted tubule. So what takes place? This proximal convoluted tubule, I told you here the filtrate is entered into proximal convoluted tubule, but in this filtrate a lot of water is remaining. Okay, actually in a human being, if that whole filtrate which is produced by Malpighian body, if really excretes from every nephron, in a person, per a day 175 liters of urine has to be excreted, but it is not happening, right? Why? Because that all excess water which is produced in this, I mean which is filtered here in this proximal convoluted tubule, it will be reabsorbed back into the body, into the blood vessels. Okay, reabsorbed. Okay, so most of that water will be reabsorbed again, and finally the remaining very concentrated, highly concentrated urine enters into this next part. This part, small part, you can see here, this is also a tubule part, but it is known as loop of Henle. Okay, or else Henle's loop you can call. Okay, what is the specialty of this? You can see here the diameter of this proximal convoluted tubule and the diameter of loop of Henle. If you observe, the loop of Henle diameter is very small when compared with PCT. Okay, proximal convoluted tubule, because here the filtrate is more produced here, but less filtrate is passing here through Henle's loop. That's why what happens here, pressure is created because more urine is gathered here, but less filtrate is allowing here. That's why allowed here. That's why here pressure is created. That's why, because of that pressure in this proximal convoluted tubule, there is a chance of more reabsorption. Okay, here in the proximal convoluted tubule, special function takes place is reabsorption. Okay, so now the urine is, I mean filtrate is passing through this loop of Henle. The concentrated urine is passing through the loop of Henle. Now later on again it enters into another tubule known as a DCT. It means distal convoluted tubule. Distal means it's originated from distant. It means it is distant from Malpighian body. It means it is a little bit far from this Malpighian body. That's why it is known as a distal convoluted tubule. Okay, so you can see here this tubular part here, very clearly you can see this tubular part is completely entangled with arterioles and venules, right? You can see here. Okay, then here in this distal convoluted tubule, tubular secretion takes place. It means the unwanted substances from these venules enters into the distal convoluted tubule, and the wall of the distal convoluted tubule secretes still more unwanted substances which all are there in the blood and into the distal convoluted tubule, I mean into the urine which is present in the distal convoluted tubule. Okay, then by the time the filtrate when reaches to this distal convoluted tubule is very highly concentrated and with completely unwanted substances and majorly nitrogenous substances. So now this enters into collecting duct. This you can see that it is collecting tubule. You can see that it is collecting tubule. Okay, so now you can see here how it is arranged. So finally this collecting tubule is formed. Okay, so now urine enters into this collecting tubule. All the nephrons collecting tubules, all the nephrons, for example I told you here calyces represents the location of nephrons, right? So here till now we have discussed about one nephron, right? So just like that, in 1.3 to 1.8 million nephrons which all are located in the medulla region of calyces in the kidney goes on functioning, and each and every nephrons collecting tubules pours urine in this conical shaped region. This is known as conical shaped part. This is known as pelvis. You can see here pelvis, right? So every nephron pours its urine through their collecting ducts. I mean all of their collecting ducts, all the nephrons collecting ducts opens into pelvis. So now pelvis is the conical shaped part which is receiving the filtrate, I mean in the form of urine. Now it is receiving the urine. So now this urine now slowly enters into ureter. Okay, So you can see here ureter at the hilus region, ureter comes out of the kidney. So in this way now urine enters into ureter. Okay, so now after entering into the ureter, what happens? You can see here again, after entering into the ureter, slowly it runs downwards and opens into urinary bladder. Okay, actually these ureters are also tubular structures, but those are closed tubes. When urine is running downwards, they just give its facility to enter through, I mean to pass through this, and they support the liquid which is coming out of the kidney. Okay, so hope you understood the structure of nephron as well as the structure of internal structure of kidney. And that's why here in the mechanism of urine formation actually divided into 4 stages. Those are glomerular filtration. Okay, I mean the filtration which takes place at the glomerulus. Next, tubular reabsorption. This is what here it takes place at the PCT. This is shortly known as PCT. It means proximal convoluted tubule, loop of Henle, and distal convoluted tubule and collecting duct. Okay, then here second stage is known as tubular reabsorption, then tubular secretion, and formation of concentrated urine. Okay, so these are the 4 stages in the mechanism of urine formation. You can see here. Okay, so I hope you understood that general structure of kidney as well as the functioning of nephron and the structure of nephron. I hope you understood very clearly. If you really like my videos, please like, share and subscribe my channel and share it to your friends, those who are in need of this.